Hello and welcome to this Australian Biocommons webinar. My name is Melissa Burke. I'm the Australian Biocommons Training Manager and I'll be your host for today. This webinar is part of a series in which we share useful information about the latest digital techniques, data and tools available to the life sciences community. Each month we hear from local and international experts who present a bioinformatics topic that will support Australians to deliver their best environmental, agricultural and medical research. You can find out more about what's happening in the Biocommons on our website or by following us on social media. Before we begin, we will take a moment to acknowledge the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. Today, our team and our speakers are joining you from the lands of the Turrbal and Yagara people in Brisbane, the Wurundjeri people of the Kulin Nation in Melbourne, and the Awabakal and Worimi people in Newcastle. Today, we're thrilled to welcome Dr. Tiff Nelson and Dr. Tristan Reynolds to the webinar to speak to us about Galaxy Australia and how this popular code-free platform is being used to support biological research. Tiff is the Australian Biocommons Genomics Research Community Engagement Lead, and she has a background in marine science, microbiology and ecology, where her work focused on the links between microbes, their environment, and the implications for planetary, human and animal health. Tiff now applies her expertise to support life scientists with data-intensive work by engaging with them to understand their computational challenges and needs, and by working with the Biocommons and our partners to ease these burdens. Tristan is the Galaxy Outreach Specialist with Melbourne Bioinformatics and Australian Biocommons, where he provides training and support on using Galaxy to researchers at the University of Melbourne and those within the Parkville Biomedical Precinct. Tristan has a research background in radio astronomy and galaxy evolution, and he has also experience on running and using Galaxy on local HPC at the Murdoch Children's Research Institute. Welcome to the webinar, Tiff and Tristan, and I'm now going to hand over to you to get it started.
Okay, thanks Melissa for the introduction. I'm Tiff and I'll be taking the first half of the presentation and then I'll be handing over to my colleague Dr. Tristan Reynolds for the second half. So to start, what is Galaxy Australia? So Galaxy is an open source software that is web accessible. And it's really being used for data intensive research. So Galaxy is developed and maintained by a global community and it's almost 20 years old. So it was really born and developed in 2005. And the Australian server represents one of the large national servers provided to researchers worldwide. So the Galaxy Australia server is offered through a team of operational, computational and funding partners, and it forms part of the National Australian Biocommons Digital Infrastructure Services group. So before I start to show you around the Galaxy Australia platform, I wanted to give you a few examples of some of the ways that researchers have used the platform to perform their research to their advantage. So the first example I wanted to show you is in relation to virus screening. So pest and disease pose a threat to national agricultural resources in Australia, and preventative measures are taken to prevent disease coming into the country. When we call agriculture, brown rugose fruit virus, really catchy. You can see some of the impacted capsicum and tomatoes in the pictures there. These viruses can cause serious economic damage with yield losses in up to 70% of stock. These types of pathogens are often spread through infected seed or plant material that are transferred through quarantine facilities or through other parts of the country. And detection is via these quarantine facilities, such as this one in Victoria, and it's done in a relatively low throughput manner using visual inspection and then wet lab procedures such as an ELISA or a PCR. When the plants arrive in these quarantine facilities, This presented an opportunity when Associate Professor Roberto Barrero and colleagues identified that this could really be moved to a much more high throughput method. He developed a toolkit that allowed viruses to be assembled in a much more efficient way. This was somewhat of a bespoke type of toolkit to understand and identify these viruses much better rather than using the more low throughput methods. Some of the roadblocks with this toolkit being adopted by quarantine facility managers was that it really was requiring of significant computational resources, and it required some command line environments. So this presented a unique opportunity for the Galaxy Australia environment to be used by in collaboration with Roberto and his colleagues to make it a much more web accessible tool. So GA via report was developed, and this toolkit was wrapped and made available in the Galaxy Australia web interface. At this time, it was developed with the Queensland University of Technology researchers and the Department of Agriculture, Fisheries and Forestries. So now what we know is that this toolkit is a primary virus screening method, and it's really facilitated this high throughput method of virus screening in the quarantine facilities and utilised by a number of different types of managers in these environments to successfully run the jobs and also detect virus early before they move around the country. We know that there's over about 4,000 jobs per month run since 2022, and that's over 100,000 jobs since it was first implemented in the Galaxy Australia platform. If you want to read a little bit more about this tool, there's a news item that was recently written about this successful deployment, and you can read about it. It'll be linked in the webinar slides when we share them after. So another example I wanted to focus on is the Abricate tool. So Abricate is the ABR in Abricate is for the antibacterial resistance. So this tool is in high use or is in use for the detection of known antimicrobial resistance and virulence genes. So it can be applied to contig or GenBank files of FASTQs to identify these types of known genes. This particular tool was authored and created by bioinformatician Associate Professor Torsten Seaman, who's at the University of Melbourne, and you can take a look at the specifics or more details about the tool on Torsten's GitHub page. This particular tool can be deployed in a number of different command line environments, but we really saw this opportunity to wrap the tool and make it available in the Galaxy Australia environment so that it can be available to non-command line users as well. This has really allowed it to be accessed by researchers that can use it in a non-coding environment. So Abricate has now been run in more than 400,000 jobs, Galaxy Australian platform, and some of these have led to publications. So some of the examples include detecting antibacterial resistance in wound-causing pathogens in various types of animals, and also to detect environmental reservoirs in things like cyanobacteria. So there are many more publications using this tool, and these are just a couple of examples that have used the tool in the Galaxy Australia environment. If you want a chance to try this tool, there is a tutorial on the Galaxy Training Network, which I know Tristan will talk about this network in a little bit more detail, but you can find this tutorial and it's highlighted there with test data to allow you to run it side by side in your own Galaxy Australia platform web interface. Again, there's another news article that you can read in a little bit more detail about this tool, and these links will be shared around with the slides for you to access later. The last example I wanted to talk about is in relation to the swift parrot. So this bird is critically endangered. It breeds in Tasmania, and it's a migratory bird that breeds in Tasmania and winters over in the mainland Australia. It has a significant amount of threat. Habitat destruction and predation are a few of the top ones, and there's only really a few hundred individuals remaining in the wild. So assembling genomic resources are really invaluable for conservation managers to understand and assist with decision making for critically endangered or threatened species. but assembling eukaryotic genomes in particular are routinely seen as somewhat complex. They're often multi-tool workflows that require coding skills. So this was a really strong point that wanted to be overcome by Professor Carolyn Hogg and Dr. Luke Silver and colleagues at the University of Sydney as part of the Threatened Species Initiative, to make these workflows more accessible to conservation managers or researchers that don't have the time or expertise in coding to really access those workflows for performing genome assembly and get access to the information needed to make critical decisions about threatened species. So the implementation of this workflow, you can see using PacBio hi-fi reads, allowed an assembled genome with a really high BUSCO score that of 97%, just almost 100% completion. And this has really paved the way for allowing these eukaryotic genomes to be performed in the Galaxy Australia platform. Again, this workflow is available with a tutorial and a how-to guide on the Australian Biocommons how-to guide GitHub. So you can take a look at that there and follow along and import those workflows into your own Galaxy Australia instance to assemble your own genome with your own data or by using test data. So now I wanted to move into the Galaxy Australia platform and just help you navigate and show you some of the environment to help you find what you might be looking for on your research journey. So when you encounter Galaxy Australia, it's accessible by the web interface. It's a platform that will display on your screen much as you can see in the screenshot there. You just register and you can sign up and start using it. If you register with your institutional email, you'll get a slightly larger portion of data quota. In this environment, you have access to thousands of different bioinformatic tools. So you can just, once you're registered, you're away. Underneath the Galaxy Australia platform, there's a number of powerful and very computational resources that really underpin, and these are located around the nation. And you can see there of different types of flavors of memory and styles to support the types of tools that are embedded in Galaxy. This is something that as a user, you do not need to manage or worry about. It's just information so that you know how jobs are shared and managed from each one of your interface or your servers once you log in. So when we encounter a Galaxy Australia interface, if we take a look on the far right of the interface, you'll see your history. I think this is much similar to managing your folders in your own directory. So these, you can have as many histories as you like, and you can name them after a particular data set or an analysis project. And I find these really powerful for helping you manage your data analysis. As anyone knows, you can get many files once you start running more than one or two tools, and this can really get out of hand very quickly. So histories, I find, are fantastic for managing your data analysis projects. You can have these histories shared with colleagues or supervisors, and you can also save these histories. And so that way you can go back and reacquaint yourself with the workflow or the versions of the tools that you used while performing your analysis. If we now go over to the far left, you can see a list of icons that will help you navigate around the platform. I'm not going to go through all of these icons. I really encourage you to go and take a look and click and read all about all the things that you can do in the Galaxy Australia workplace. But what I will focus on is one that is very useful, and that is the upload tool. So this is usually your starting point. You might create a history and rename it, and then the next thing you'll start to do is uploading your data. You can upload your data in a number of ways. Most commonly, people might upload from their local environments. So you can choose individual local files and upload them into your history. And then another feature which I find incredibly powerful, especially if you have a large collection of data files that you're going to be analysed and processed together, you can upload files as a collection. So this is really useful for keeping them together. And that way, when I name that collection of, in this case, it's only 2 you can see in the list here, but you might have more than 50. You don't need to then keep tabs that you've included them all in your analysis. You can simply refer to them as My6 or whatever you decide to name them. And it really helps you easily perform your downstream analysis steps. You can also upload your files from remote destinations. So this includes some valuable destinations where you might want to access known data sets that you can use in testing workflows or comparing workflows. It also includes a shared cloud spaces where you might keep your data. And this is really efficient in terms of limiting transfer from and to your local environment. If you want some more information or to really discover some of the efficiencies in terms of uploading your data, you can see the top tips video on uploading your data that's on our YouTube channel. And again, that's going to be linked in our webinar slides. The next thing I wanted to show you is, if we move to the left, not the far left, the next panel over, you can see a list of the tools. When you click on any of these sections, you will get further information about the types of tools that you might encounter under a given category. Something that I find incredibly powerful is to then use some of the other categories to summarize some of the tools you might be looking for. You can obviously use the search bar to find a tool when you know the name. So you might know that you need the abricate tool, but what often happens is you want to perform an analysis, but you don't know the tool that exists. And so in that way, these other types of ways of searching for tools can really facilitate you finding the tool or the analysis pathway that you're needed to perform the task that you're looking for. So the EDM categorization, and EDM really stands for Data Analysis and Management Ontology. This can be broken down into operations and topics. So if we take a look at operations for a minute, you can see that you might come to the platform and you know you need to trim off some primers or shorten some of your FASTQ reads. You might then go to sequence trimming, and this will give you a list of options of particular tools that can perform this type of technique. And this really enables you to not only find a tool that you might need to perform your analysis, it also allows you to find a tool that could replace another tool if that particular tool isn't available for some reason or the version's slightly not working the way you'd like it to. So I find this incredibly useful way to search. The other way is through the topics. You might be new to a particular field or have your first data set in the microbiome space, for example. So you have that data set and you really want to focus on where do I start? And this might be another avenue for you to work through. And the last one I wanted to like watching that tool feature. So from the search bar in the tool panel, you can then see that there's arrow pointing down. You just click on these arrows, and this provides you with a way to filter by any type of name or operation to find particular tools of interest. For example, in this case, I know that I need a tool that trims, and by filtering by naming of trim, I'm able to see 20 different tools that perform trimming, and I can scroll through this list of tools to try and find more information if it trims, reads in a BAM file or reads in a text file, and I can find the type of tool that I'm really looking for. So again, this is just another powerful way to find something that you're looking for without really knowing the answer necessarily before you begin. Now I'm going to, so I've shown you through a couple of the features of searching for tools or analysis workflows when you don't necessarily have all the information. And a new feature of Galaxy Australia are the labs. So the labs provide a curated view into a subset of tools around a domain area. And we have created these labs in particular domain areas. And what you're looking at in the movie on the screen is the Genome Lab. So the Genome Lab provides a number of tools that we know the community is using in high rotation, and also allows you to use particular workflows and tools for steps such as genome assembly or genome annotation. We have a number of different labs as well as not just Genome Lab. We also have the Proteomics Lab, Microbiome Lab, and the Single Cell Lab, but the Genome Lab is somewhat more mature than some of the other labs. And you can see in the movie as it's moving down, you have helpful tips to allow you to answer questions, particularly valuable if you're new to an area of navigating a data analysis field or domain area to try and find areas of interest or workflows that you'd like to navigate through. Now, this particular genome lab is available at genome.usegalaxy.org.au. You can also find all of the types of labs by clicking on the hexagon in the top banner of the Galaxy Australia once you're logged in. It's important to add that even though you're in the genome lab and it provides that middle panel of curated tools and workflows and sort of links to tutorials, you still have all the tools available and the same amount of compute and tools available to you underpinning the Galaxy Australia server. You're just really getting a more curated view into the area of interest in that analysis pathway to facilitate your learning. Another feature of Galaxy that I want to highlight before I move over to Tristan is accessing particular specialised tools or additional storage or request of tools. So moving from right to left, you can see if you scroll down in the middle panel on the main site of Galaxy Australia, you can see that there's a way to request additional storage. You simply go to request now and fill in the form. There's a way to request tools and data sets. If you really need a particular tool and you can't quite find what you're looking for, you can go in and access a tool shed or a library of tools that are available that you can see if it's available to be implemented or deployed in Galaxy Australia. And then in the national Australian Galaxy Australia server, we also have a number of specialised tools that have been deployed in Galaxy Australia through engagement with the Australian life science research community. So we know that these tools are of particular interest and required by the research community. And for this reason that we've made them available in the Galaxy Australia platform. Some of them you might be familiar with. AlphaFold 2 is predicts protein structures from nucleic acid sequences, or protein acid sequence, I should say. FGenes H++ annotates genomes automatically. Cell Ranger performs a single cell RNA-seq analysis, and Diane is a proteomics data analysis tool. You can apply for these. It's a short form and it basically allows us to check that you meet the licensing agreements that we have with each of these tools. And these tools have been made available for the Australian audience to really value add to their research analysis pathways.
Great. So I will now tell you a bit more about how Galaxy supports reproducibility, and then after that, about the training and support that's available with Galaxy. Oops. Sorry. There we go. Make sure that I can actually navigate my slides. Sorry, too far. So when you're doing your research analysis, and particularly when you have large numbers of samples, rather than analyzing each of them individually, you'll often want to go and create workflows or pipelines so as to automate your analysis. And this has many benefits. It makes your data analysis more reproducible. It also streamlines your data analysis. And there are many ways you can do this. There are simple ways using just scripting languages such as Bash, Python, R, C, whatever language you want. There's also a bit more advanced pipelining languages such as Nextflow or SnakeMake, which is a Python-based version. But in all of these cases, you're working with code, and so kind of script files, kind of as the one shown here. Galaxy also has the ability to create pipelines, and in Galaxy they're called Galaxy workflows. And this goes and allows you to go and build up in a graphical way and link the outputs and inputs between different tools. So this way you can go and create any sort of a complex workflow you want, and then use that to analyze all of your data sets in exactly the same way. And when you create a workflow, all of any parameters or settings that you specify for each of the tools in your workflow will be saved. So it means that you have consistency for every time you run the workflow. And just shown here is actually one of the workflows used in the Swift Parrot study that Tiff was talking about earlier. Yeah, so different benefits are that you're performing the exact same analysis. Although the parameters are set and are fixed within the workflow, if you're, say, for one of your samples, you need to modify some of the settings, you can manually modify different tool parameter settings for individual workflow runs. So there is some flexibility there that you don't have to go and recreate the entire workflow when there are minor changes. And also a benefit is that your analysis will be more streamlined if you use a workflow in Galaxy, because Galaxy will automatically submit and run the different steps in the workflow as soon as all the required inputs are available. So it means you don't have to wait for step one to finish before you run step two, and you might have a bit of lag time there if you don't realize that step one is finished already. As Galaxy goes, it's like step one's done, all right, submit and run step two. And Galaxy workflows can be shared and published, which I will talk a bit more later on. But that is a very powerful functionality. So there are multiple different ways that you can create or use workflows within Galaxy. The first one, this goes back to the sharing and publishing of workflows, is that you can import published workflows from a few different sources. From the sites as Tiff showed earlier, but also there are two dedicated repositories which include Galaxy-based workflows in addition to Nextflow and other workflows. These are Workflow Hub and DocStore. And in both of these cases, this allows you to get inspiration. If you're not sure exactly what you want your workflow to do, you can look for something that does something similar, and that can be your starting point to build up your own workflow. You can also use this for if you're referencing a published paper and you want to reproduce their results. If they have published their workflow, then you can grab their workflow and reproduce exactly what they did, which is powerful for, yeah, that reproducibility of science. You can also extract a workflow from your history. So you can go and run tools individually, particularly if you're going and testing and trying to figure out which tools and what settings you want to go and use. And once you've gone and completed your analysis for a single test data set, you can then extract from that history all the tools that you ran, how they link together, what settings were used, and then Galaxy will automatically generate a workflow for you. And you can also, if you run a tool multiple times, you can remove some of those times you ran a tool from the created workflow. So if you ran a filtering tool 3 times, but you only want to use one of those filtering settings, you can unselect the other 2 times you ran that tool. And you can also, if you already have an idea of what you want your workflow to do, you can go and create a workflow from scratch. So you can just go and have a blank canvas and then just select tools from the tool panel and add them to your workflow canvas, and then just drag to link the outputs of certain tools to the inputs of other tools. So there's a lot of flexibility on how you go about creating your workflows. There's a few other kind of more high level attributes of workflows which are important. One thing is that workflows do have version control. So every time you save your workflow, there is a saved version that you can always go back to if you at some point decide, like, oh, I actually want to see I want to go back to my workflow when I was working on it last week or last month. You have that full version control history. And then if you're publishing or sharing your workflow, 2 important things to include are a license, so that lets people know how they can actually use your workflow, and also to list all the credits, all the contributors who contributed to actually creating that workflow. So make sure everyone actually gets the recognition for the work that they did. And particularly if your workflow, you're publishing it, if someone else uses it, means that everyone goes and gets that recognition and citation. You can also, in workflows, if you've created a workflow to do some specific little task, you can put that inside of another workflow and what's called a sub workflow rather than reproducing that handful of steps to do, say, a specific filtering task, which is what this example does, you can go and include that inside of another workflow as a single step. So that's also a nice flexibility that you can go and reuse already existing workflows rather than having to every time start from scratch to recreate your workflows. And some recently added within the last, I think, year or so functionality are workflow comments to increase the readability of your workflows. So a few different types. There's just text, so just basically a text box, so you can just add more large-scale comments. Markdown, so it uses the Markdown text format, and that allows you a bit more flexibility and allows you to go and say use bullet points and headings within your comments. Frame comments, which are my favorites, they allow you to go and take individual tools and group them together, as shown here in this workflow. And that is very useful and very powerful for increasing the readability of your workflow, and particularly if it's a very complex workflow, when you're then sharing and someone else is trying to understand what the different steps of your workflow are designed to do. And also, once you've grouped the tools together, you can move and rearrange the workflow frame, and it will move all of the contained tools at the same time. So it means it makes it easier to go and edit and rearrange the layout of your workflow. And there's also a freehand pen if you want to go and make just say small diagrams or your own just handwritten with your mouse comments. And this just a nanopore processing workflow that I found on Workflow Hub just kind of shows that the powerful nature of using workflow, the frame comments in going and grouping together the different tools. So you can much more easily go and follow, so, okay, what is this group of tools designed to go and do and see the logical connections between things. When you actually submit to go and run a workflow on Galaxy, you will get a workflow invocation, and this will go and show you a nice diagram layout, which will show you the progress of your workflow as it progresses. You can see each tool, how it links to other tools, and you can see as each tool completes running. So as it goes from being gray, so queued waiting to run, orange running, and hopefully if it succeeds, it'll turn green. If it turns red, then you've got a clear indication that that tool has failed and needs to be looked into. And you can view all past workflow invocations within Galaxy as well. So there are many ways that you can share and publish your workflows. The easiest way is you can just download the workflow as a file, which you can then share with anyone, and they can then upload into any public or local Galaxy server. You can't really interact with that downloaded file on its own. You need to put it into Galaxy to interact with. But then specifically within Galaxy Australia, you can share workflows with other users. So you can share with a user by their email address. You can also share via a link, and you can also you can publish it so it will appear in the public workflows tab in Galaxy Australia. Then anyone using Galaxy Australia can search for and find and use that workflow. You also have the ability, if you have your own website, that you can embed a image of your workflow in your website. So that can be a way to go and increase visibility of any workflows that you create. And as I mentioned earlier, you can also publish your workflows to Workflow Hub and Dockstore, from which you can also, you can or other people can download directly, import directly into Galaxy Australia or another Galaxy server if you're working with people overseas. Now, what Galaxy training and support is available? There's a whole bunch of things that are available. In terms of looking more globally, so for all around the world, for anyone using Galaxy, there's the Galaxy Training Network, which people contribute to from anywhere around the globe. And this provides over 300 different tutorials on how to do basic Galaxy functionality, but also how to perform different types of analysis within Galaxy. which you can just do a search, but also they're grouped based on methodology and also scientific field. And it's just showing the range of methodologies and scientific fields that are covered, and it just shows the range of different topics that people are using Galaxy to do science with. Just for an example of one of the tutorials and some of the additional resources that are available, some will have slides associated with them, either on the going a bit more detail into the type of science being that the tutorial covers. They will also include data sets, so that's the actual test data or sample data to go and use for running the tutorial. Some will have workflows, so basically taking all the tools that are run within the tutorial and turning those into a workflow, which can give you an idea. Another way to get an idea of is like, oh, I'm trying to do a workflow to do RNA-seq analysis. Well, there's RNA-seq tutorials, which you can go and get a workflow from there to go and be a starting point to go and build a workflow that does exactly what you want. Some will also have recordings, which is actually, it'll be someone has gone through and recorded and explained going through and actually carrying out the tutorial, which can also be a handy thing if you want to actually go and see exactly how to go through the tutorial. Coming up week after next, so from May 12th to 16th, there's the Galaxy Training Academy for 2025. So this is a week-long asynchronous, so it's an online training that there's set tutorials for each day of the week from the Galaxy Training Network. And the power of doing the tutorials during the GTA is that there are instructors who you can go and contact over Slack if you have questions about anything going wrong with the tutorial or just questions of get more in-depth understanding of the tutorial, but also of the actual science being that Galaxy is being used for in some of the different tutorials. So if you haven't registered and I think that you would find this useful and interesting, I encourage you to register. And an important point, there is no cost to use it. And because it's a global event, no matter what time you're carrying out tutorials, there will be someone online who can go and answer your questions. Obviously, if you're carrying it out overnight in Australia, you won't get support from people within Australia, but there'll be people online from either from somewhere else around the world who will likely be able to answer your questions. Moving more locally, the Australian Biocommons has a Top Tips from Galaxy Australia YouTube channel, which has a number of videos with covering different topics of Galaxy and different Galaxy use, which Tiff also mentioned earlier about, I think it was Abricate. So for more details on different topics about Galaxy, encourage you to go and take a look there as well. Now, specifically, if you're using Galaxy Australia, there is support that you can request and assistance you can request from the Galaxy Australia team. So we have a help desk, and you can request help with various things. One of the main ones is support if your job fails. Now, everyone hopes the job never fails, but reality is your jobs will sometimes fail. If they do, the output in your history will turn red. And if you click on the name of the error data sets, it will expand and there will be a bug icon at the bottom of it, which if you click on that, it'll bring up the error report for your tool run. And when you're submitting a help desk support request, if you include the error report, it will help the Galaxy Australia team in troubleshooting and solving your issues. So that's an important thing to go and include when you are submitting support requests. As Tiff also mentioned earlier, there's also a number of other things you can request support from the Galaxy Australia team, including getting access to special tools such as AlphaFold 2, FGen's SH Cell Ranger, and forgot to include on the slide, but also Diane. Also requesting new tools and data sets if the tool you're after isn't installed in Galaxy. And you can also request additional storage if you're carrying out a project where 600 gigabytes is enough, and for a short time frame, you need a bit larger, say a terabyte or a bit more. You go and request that additional storage so you can actually carry out your analysis. Now, this isn't intended to be for long-term storage to go and just continuously increasing your storage in perpetuity, but so you can go and carry out your research over a few month time frame. And in terms of installing new tools and data sets, the Galaxy Australia team highly encourages feedback and input on what tools that are not in Galaxy are ones that researchers will find useful. And because it takes a bit of time to go and actually install tools, all requests that come in will be prioritized based on the level of interest in the various tools. But for the most part, there's not been any case where there'll be very few cases where it's like, no, that tools can't be installed. Almost all cases, tools will be able to be installed, but it may go and take a little bit of time depending on the capacity of the team at a given point in time. Potentially also of interest is Galaxy has a training infrastructure as a service. So if you're running a training event, you can submit a request for a allocation in our training infrastructure as a service, which goes and provides a priority queue for running jobs for your training event participants during your training. So as rather than having to submit jobs to the normal queue, your jobs will go to a special queue for training. And to wrap up, I want to give a shout out and highlight this is the Galaxy Australia team. So if you're using Galaxy or if you're reaching out for help or support, these are the people behind the scenes who are making sure that Galaxy is running. As you can see, they're from a range of different institutes and different affiliations. So we're not all based in any one location. We're spread out, which means we're constantly interacting with lots of people, and we obviously go and support the entire community. And just finally, just to go and show that Galaxy only functions because of all the operational and computational partners and the funding we have. So yeah, because Galaxy Australia is part of the National Australian Biocommons infrastructure and is made possible through the collaborative efforts of multiple operational and computational partners and funding from the University of Melbourne, Bioplatforms Australia, NCRIS via Bioplatforms Australia, and the Queensland government. Yeah, it's only thanks to everyone listed here that Galaxy keeps working and being able to support everyone's research. And that's everything. Thank you.
Thanks so much, Tiff and Tristan, for that look into Galaxy and how it's being used for research and a little peek at some of the interactive features that make it such a powerful tool. We have time now for some questions, so if you do have questions, please type them into the Q&A box and we'll do our best to answer those for you. One question that is coming up is about the funding of Galaxy and what this means for researchers. Tiff, I wonder if you could talk a little bit about that.
Yeah, thanks, Melissa. Yeah, so I guess the way to explain it is that Galaxy Australia, as a user or as a researcher, is fully subsidised. So you can approach the platform and use it without any cost to you. And the operational and computational and infrastructure partners have significant or have funding to support the Galaxy Australia platform. So there's no cost to you. It's fully subsidised, but in the back end, the computational and operational partners really make Galaxy Australia to the life science research community to perform their analysis.
Thanks, Tiff. And I'll just add that the same is true for the Galaxy training tutorials that Tristan was talking about. Those are made available at no cost to you by the global Galaxy community. And there's a wealth of different tutorials in there for you to explore. The next question well, there's a few questions that are similar and they tend to be around, can I use Galaxy for X? For example, plant microbiology or eDNA. How can people find out what types of analysis Galaxy is useful for?
Yes, I think the best thing is to go and start exploring. Many people approach the platform by making their own workflows, or many start by piggybacking on other workflows that have been developed. You can see in the platform interface where Tristan was showing where the workflows you can find published workflows. So not only can you go to the Galaxy Training Network, if you have a colleague or a paper or any published workflow in an area, you can really go and bring that into your own history, find some test data or request some test data, and then apply that workflow to your own analysis project. That is incredibly valuable. The other way that you can learn more is through exploring using those EDEM operations or domain areas of interest. I think we will be actively developing some areas, including eDNA, in the next year or so. What we're seeing with the labs is that that provides an avenue for researchers to come in with limited experience in a domain area and really have no knowledge of what tool they want to use, but be able to peruse that middle panel to find a workflow or a process and then apply it to their own data. So I guess the short answer is just go and have a look on the Galaxy Training Network, or also in the Galaxy interface, to try and find some of the tools using those advanced search features that I mentioned.
Thanks, Tiff. And it's true that there is such a variety of types of data analysis that Galaxy can do. It's fairly likely that there will be tools available in your domain for you to use.
That's right.
The next question we have is, Galaxy Australia only open to researchers from Australia? This particular person is in New Zealand, and I know there's a few other international people on the webinar as well.
It's not. It is open to anyone that can has a internet connection basically and can access the web. So you only get a small perk with the institutional Australian institutional email address when you register. You get a 600 gigabyte quota, whereas users will get a smaller quota when they register. Bearing in mind though, we do make available to others that you can request for further storage. So you can request for short periods of time where you increase your data storage. You might have a project where you have a need for, let's say, 1 terabyte or 2 terabytes because you're running a genome assembly project and you just need access to that for a couple of months. We can provide increased data storage for periods of time, and then we can also allow those users to have access to that for performing their analysis.
Thanks, Tiff. And to add to that as well, Galaxy Australia is part of that global network of comparable Galaxy platforms. So if you are based in Europe, there is Galaxy Europe that you can use, pretty much identical tools and resources on there, and the same with Galaxy Project, which is based in the US. There's also a number of other national servers across the world as well. So there will be a Galaxy that's suitable for you somewhere.
I guess I was just, I know Tristan spruiked this, but I saw there's another question in the chat about the Galaxy Training Academy. So this is something like an asynchronous sort of training event, and this is probably the perfect example to get access to experts in all sorts of domain areas to really apply their knowledge to the Galaxy Australia environment, or actually it's a global sort of event. So what is really useful about that is you have access to not just sort of bioinformaticians or biologists, but you can also talk to some of the tool wrappers and people that have a really intimate knowledge around how Galaxy Australia works. So I encourage anyone to block out some time in that week that it's being held. It's held across the whole week at a sort of like all sorts of hours, because it's a global platform and a global sort of open project. Come along and you will learn something, and you will be able to, even if you run a tutorial because you want to get into Galaxy Australia or learn more, that is the time to do it, because then you can be asking questions as you're going, and almost always someone's on hand at various times of the day and night to answer your question. That is a really niche question to your research. You can often get help during that week. So that is a great time to get into Galaxy Australia and run it. You know, with your colleagues, maybe in a room together with your computers. It can be really valuable to learn really quickly how to use the interface.
I think we will leave it there for today. So I just have a few things to wrap us up. If you'd like to find out more about what's happening at Biocommons and upcoming training that we have on offer, you can subscribe to our newsletter. You can also follow us on Blue Sky, LinkedIn, or on our YouTube channel. The other things that you might want to have a look at are the domain pages that describe how we're engaging with different community groups across Australia, and you can browse upcoming webinars and workshops on our website. So that just leaves me to thank Tiff and Tristan once again for a great introduction to Galaxy and how it can be used for life science research. As we leave, I'd like to acknowledge that Galaxy Australia is part of the National Australian Biocommons infrastructure, and that Australian Biocommons is enabled by NCRIS via Bioplatforms Australia funding. Thank you to everyone for joining us. We hope that you've enjoyed this webinar and that we will get to see you again soon. But until then, goodbye for now and enjoy the rest of your day.